Chapter 4 is on the anatomy of prokaryotic and eukaryotic cells. So we will mostly focus on prokaryotic cells in this class, mainly bacteria. You can kind of, when you hear the word prokaryotic, put bacteria in that place, or vice versa, right? Bacteria are prokaryotic cells, and that's really our focus because we're not going to focus much on the other group of prokaryotic cells, which is archaea. So for the most part, you can kind of interchange prokaryotic and bacteria, and we're going to kind of do a compare and contrast to eukaryotic cells in this chapter. So I told you back in chapter 1, kind of the definitions and meanings of prokaryote, eukaryote. So hopefully you know that already. And let's watch this little video. Oh, we need sound.
I'm Jonathan Proper, CEO of Jobs.com, and I'm here to give you the naked truth about laundry.
If you had to think back about the most exciting day you ever had in the science classroom, which day would that be? Now, looking back through the years, we have a few. The time we participated in an earthworm dissection, the time we took apart an owl pellet, the osmosis eggs, all of the fruit flies and genetic experiments. Oh, I could go on, but I will never forget one day in my 9th grade science class. My teacher brought in pond water, and I put one drop of pond water on a microscope slide and saw the most amazing thing ever. I saw an amoeba. A single-celled amoeba on that microscope slide, and I was forever stuck on science from that point on. Because I could not believe that this little cell was there, alive on this slide, still eating, because that's what amoebas do a lot. But to imagine that every person is actually made of billions of cells. Of course, not amoeba cells, but animal cells. Billions of animal cells. That's fascinating. In fact, it really makes you reflect on some of the incredible statements of the modern cell theory. The modern cell theory includes the following. First, that the cell is the smallest living unit in all organisms. And second, that all living things are made of cells, one or more cells. The amoeba I observed was a single-celled organism, so unicellular. Humans are made of many cells, so multicellular. And third, all cells come from other pre-existing cells. You know, cells have their own little world inside them. They carry genetic information, they can divide, many have functions and processes that their organelles, structures inside them, can take care of. On our planet, we can divide cells into two major groups. As a cell, you're either a prokaryote or a eukaryote. Bacteria and archaea are prokaryotes, but everything else, plants, animals, fungi, protists, are eukaryotes. Both prokaryotes and eukaryotes have genetic material. Both have cytoplasm. Both have ribosomes, which are small organelles that make protein. Both have cell membranes, which control what goes in and out of the cell. But what makes them different is a big deal. Prokaryote, the pro rhymes with no, and they have no nucleus, which holds the genetic material and controls the cell's activities. Prokaryotes have no membrane-bound organelles. Membrane-bound organelles are fancy organelles like the nucleus and mitochondria and Golgi apparatus. Eukaryotes, the U rhymes with do, they do have membrane-bound organelles. So now you may be wondering, well, what do the organelles do? What are their functions? Well, you know our style. We love our science with a side of comics. So we want to take you on a tour of the ride of your life, into the inside of a cell. Now, to start our trip, we're going to have to get through this cell membrane, also called a plasma membrane. It's selectively permeable, which means it'll only let certain select materials in and out, but by doing so, it keeps things in the cell stable, also known as keeping homeostasis. And we have an entire video on just the membrane itself, which is found in all cells. But for now, we're just going to have to squeeze through this protein in the membrane. Now, inside the cell, we find ourselves in this jelly-like material called cytoplasm. It surrounds all these internal cell structures, and you'll find it inside both prokaryotes and eukaryotes. Now, organelles that are floating around in the cytoplasm can have more support than you might think. Cells contain a cytoskeleton, which is a collection of fibers that can provide support for the cell and its organelles. And the cytoskeleton can even play a major role in cell movement. The cytoskeleton actually deserves its own video, because it's very complex, and its organization can vary depending on what kind of cell you're looking at. Moving through the cytoplasm, let's start with ribosomes. They are not membrane-bound organelles, and they're going to be in both prokaryotes and eukaryotes. They make protein, which is really important because that's what so much of genetic material, DNA, codes for, protein. Ribosomes can be free in the cytoplasm, but they can be attached to another organelle too, which we'll talk about a little bit later. We're now going to focus on organelles that will be membrane-bound. So we're going to be focusing on organelles that you would find in a eukaryote cell. This takes our travel to the big boss, the nucleus. Now in eukaryotes, it holds the genetic material. Genetic material as in DNA, for example. Both prokaryotes and eukaryotes have DNA, but if you're a eukaryote, you have a nucleus to put it in. And the nucleus controls the cell's activities, and inside it, it has a nucleolus, which is where ribosomes can be produced. Attached to the membrane of the nucleus, or nuclear membrane, you will find the endoplasmic reticulum, ER for short. It does a lot of processing of molecules for the cell, like protein folding, and it's highly involved in actually transporting those molecules around, like a highway. There's a rough ER, which has ribosomes attached to it, making it, as you can imagine, rough, and then smooth ER, which doesn't have the ribosomes. Rough ER specifically tends to be involved with protein producing and transporting, because remember that ribosomes make protein. Now, molecules that leave the ER can be sent away in vesicles that actually pinch off the ER themselves. Now, smooth ER has many additional roles, including detoxification, which is one reason why your liver cells tend to have a lot of smooth ER. And another additional role of smooth ER is that it can make some types of lipids. Next, the Golgi apparatus. It's the ultimate packaging center. It can receive items from the transport vesicles that pinched off the ER. It has enzymes that can modify molecules it may receive and sorts the material it receives as well. It can determine where to send those molecules, including some that may eventually be sent to the membrane, so they can be secreted, which means items that can be sent out of the cell. So with all that's going on in here, you might start to wonder, what's powering this thing? The mighty mitochondria, or mitochondrion, if you're just talking about one. They're like power plants. The mitochondria make ATP energy in a process called cellular respiration. Now, it's not the type of power plant that you might think of. It runs on glucose, which is a sugar, and it needs the presence of oxygen to efficiently make ATP energy. Now, at this point, we need to mention that eukaryotes are not a one-size-fits-all. Animal cells can have differences from plant cells. We have a fork in the road here. For example, plant cells not only have mitochondria, but they can also have these awesome organelles called chloroplasts. Chloroplasts actually make glucose by using light energy in a process known as photosynthesis. They tend to even have a green look to them because they have a pigment that captures the light energy and it reflects green light. Now, both plant and animal cells can have vacuoles. Now, vacuoles can have a lot of different functions, but many types act as storage of materials. Plant cells can have one large vacuole called a central vacuole, while animal cells can have smaller several vacuoles. Now, remember how we already said that all cells have membranes? They do. But plant cells additionally can have a cell wall, which is a layer that offers additional protection and shape maintenance that animal cells do not. Okay, now how do we get out of this animal cell that we've been in? Well, we could get out like a protein would. So if we were a protein, we would only be made because of instructions from DNA. And remember that in eukaryotes, DNA is found in the nucleus. We would be made by a ribosome because ribosomes make protein. The ribosomes could be attached to the rough ER, and the rough ER highway would provide a vesicle to send us to the Golgi apparatus where sorting can take place. And, if we're tagged for being secreted, we could be sent off through a vesicle from the Golgi to the membrane. Out we go! Now just keep in mind that in our quick tour, there are still so many more awesome organelles found in lots of different types of eukaryote cells to continue exploring, so to the Google for more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, oops, I forgot this was on the side. All right, so I love the Amoeba Sisters. You'll probably see a lot of their videos throughout this course. That one was a little long, but it's a good introduction to cells, and kind of, really focuses on what we're going to focus on in this chapter. Although we will give more attention to prokaryotes, and that video did give a little more attention to eukaryotes. So here's a comparison overall of prokaryotic versus eukaryotic cells. We have a homework assignment that you'll be doing, that you'll be creating a Venn diagram. So some of this information here, maybe all of it, should go on your Venn diagram most likely. So a lot of these were pointed out in the video. Both cells, of course, have DNA. It's just different in how it's arranged. Prokaryotes obviously don't have a nucleus around their, they don't have that nuclear membrane around their DNA, and they only have one circular chromosome, versus eukaryotes that have paired chromosomes that are in a nucleus. There's no histones in prokaryotes. There's no organelles. Now we're talking about membrane bound. That's what was nice about the video too, that they did distinguish that ribosomes are not organelles. They're not membrane bound organelles. And both prokaryotes and eukaryotes have ribosomes, although there is differences between them that we'll look at later in this chapter. Bacteria have cell walls, except for one group of bacteria called mycoplasmas. Otherwise, all bacteria have a cell wall made of the peptidoglycan. Eukaryotes, some groups of eukaryotes have cell walls and some don't. You might know, and we looked at this a little bit in chapter 1, fungi have cell walls, algae have cell walls, and plants, which we won't really talk about in here, but plants have cell walls. Animal cells and protozoan cells do not have cell walls. So it does vary for the eukaryotes whether they have a cell wall, and if they do, it's usually made of some kind of polysaccharide. For archaea, remember that they are also prokaryotic, but one thing that they do differ from bacteria is that they don't have peptidoglycan. They have something they call pseudomurine for their cell wall, if they have a cell wall. Some of them actually don't have a cell wall at all. And then as far as division goes, eukaryotes divide by mitosis, prokaryotes divide by a process called binary fission. For the size, shape, and arrangement of bacteria, so the average size is in the micrometer range, and here they're kind of giving a diameter and a length, so this is more focused on rod-shaped bacteria, but in any case, if they were cocci, then you would just have the diameter number, right? They're in the micrometer range. Most bacteria are monomorphic, meaning they only have one shape. They're either cocci or rod or spiral, and they can't, have multiple shapes. But there are a few bacteria that we call pleomorphic that can change their shape, that can have multiple shapes. So here's the basic shapes of bacteria. You have bacillus, or rod-shaped, and you can have a single bacillus, or sometimes we call them a regular bacilli, just meaning it's just a regular rod. I think even sometimes they call it straight, a straight rod, just a regular rod-shaped cell, versus a coccobacillus, which is a more short, plump, kind of rounded rod. And then you have coccus, or cocci, which are spherical, and they're showing a diplo there, a pair, but they don't have to be in pairs. They could be individual pairs, groups of 4, which are called tetrads, chains. I don't know why they're showing a pair there. They probably should have just showed an individual, but that's fine. And then for spiral-shaped bacteria, you have the spirillum, you have Vibrio, which is a curved rod, kind of. I mean, it's in the spiral-shaped, but you can see it only kind of has one bend to it versus the spiral, or the spirochete, which is a corkscrew-shaped. So speaking of arrangements, so diplo means pair. So you can have Diplococci, you can have Diplobacilli. Cluster, you really only find with cocci. You don't have such a thing as Staphylobacilli. Okay, so clusters is an arrangement really limited to cocci. And so clusters of cocci is called Staphylococci. Chains is strepto, and you can have both cocci or bacilli in chains. So you can have streptococci or streptobacilli. And then groups of 4, this is usually just seen in the cocci tetrads, and as well cube-like, which is groups of 8 called Sarcinae, is usually with cocci. Okay, so that's what they're demonstrating here. All of the possible arrangements of cocci specifically. Diplo, strepto, tetrad, Sarcinae, and then staphylo being just a large cluster. For bacilli, if they're just by themselves, generally, they're called single. If they're pairs, they're diplo. If they're chains, they're strepto. And then this isn't really an arrangement, it's actually more of a shape, but just showing you again coccobacilli. But what you'll notice on these pictures A and B here, they actually used the same picture. But you'll notice even when they're showing a single bacilli here in the electron micrograph picture, you can see that some of the bacteria are single, but you also have diplo. And then you see with the coccobacillus picture here that I think mainly, I can't see the divisions between these, but I think these are all diplos. Oh, there's a single one right here, right? But so bacteria don't just have to have one arrangement. When you look under the microscope at a bacterium, you might see single, diplo, and strepto, right? So don't think that they can only have one arrangement. So one thing you're going to see throughout this course is that scientific names are very important, and it's very important to write them the way they should be written, which we covered a little bit in chapter 1. That when we talk about a genus name of bacteria, that it should be capitalized and it should be italicized, or if it's handwritten, it should be underlined. And that's important because you have a genera of bacteria called bacillus, like Bacillus anthracis, which causes anthrax. But you also have a shape of bacteria called bacillus. So how do you know which one you're talking about, the scientific name or the shape? Well, it all comes down to how it's written. And that's why that's so important. So here's a question about how could you distinguish streptococci versus staphylococci under the microscope. So we will answer this separately. So moving on to look at the structure of a prokaryotic cell. What we're going to do for the rest of this chapter pretty much is work our way through this diagram, starting from the outside, working our way in. So I will divide up the lecture into multiple parts. So I'm going to stop here and then we will pick up with part 2 of the lecture here.